topic for this session is the morphology of bacteria. So before going on with the morphology of bacteria, let us know what is microbiology. Microbiology is the study of microorganisms that are too small to be seen with the unaided eye. If the object has a diameter of 1 mm, minute details of the object can be perceived with the naked eye. But if the object has a diameter of less than 1 mm, human eye cannot perceive it. So the microorganisms which are less than 1 mm in diameter are included under the broad domain of microbiology. The examples of these organisms can be bacteria, viruses, lower algae, lower fungi, protozoa, etc. Now, the classification of this organism was, before these microorganisms were included under plants or animal kingdoms. But due to certain unsatisfactory characteristics of these organisms, they have to be included under the third kingdom, which is called as Protista. So, the three kingdom classification was introduced by Carl Weiss, where Protista was included, and under this Protista, the microorganisms were included. So the kingdom Protista can again be classified into two. That is, one is prokaryotes, the other one is a eukaryotic microorganism. Prokaryotes are nothing but the organism with a primitive nucleus. So this prokaryotic organism can further be classified as bacteria, which are true bacteria, and the other one is a archaebacteria, the modified form of the bacteria or the primitive bacteria, you can say. And the next classification is eukaryotes. Eukaryotes includes the true nucleus microorganism, which are nothing but plants, animals, or the fungi. So the three kingdom classification was plants, animals, and the Protista. The Protista is again divided into the prokaryotes and the eukaryotes. So prokaryotes, we have bacteria, which is a true bacteria, and the primitive bacteria, which is a archaebacteria. The next is eukaryotes, having the true nucleated organism. Now coming to bacteria. Bacteria are the heterogeneous group of microorganisms which are of very important to human beings. So these are prokaryotic organisms because they have a primitive nucleus, which means they do not have a true nucleus. And the genetic material is usually present in nucleolus, but not in a particular true nucleus which is present in the eukaryotes. Then these organisms are mostly unicellular in nature, that is single-celled organism, which can also be present as unicellular or in a colonial form, that is in the form of colonies. And the size of this organism is very minute, and they are usually measured in micrometers. The other units being angstroms or the nanometers. So the smallest bacteria known to us is about the size of 0.2 to 1.5 micrometer in diameter and 3 to 5 micrometer in length. Now the study of this microorganism was not possible under the invention of the microscope. So microscope is a very important tool that helps us to know about the invisible group of microorganisms. And the study of different types of microscopes to study different types of organisms in different conditions is called as microscopy. So it was Antony van Leeuwenhoek who gave an insight to the invisible world of microorganisms, as he had the hobby of grinding lenses and combining them to produce magnification. So microscope is a tool, very important tool, that help us to know about the invisible group of microorganisms. And the most important thing about the microscope is that it provides us a magnification. Magnification is nothing but the enlargement of the object to an extent that we are able to visualize it. So this magnification is obtained with the help of different types of lenses that are combined together to enlarge the object to an extent that we are able to see. So there were different scientists who came up with the preparation or invention of different types of microscope, and it was Antony van Leeuwenhoek who was able to give us an insight to the invisible domain of microorganisms. So he had the hobby of grinding lenses and observed different natural materials under his microscope. So during his studies, he was able to see the raindrop having the minute organisms that were moving in the raindrop. So he called them as animalcules that laid a foundation to a broad domain of microbiology. So it was Antony van Leeuwenhoek who had given an insight to the broad domain of microorganism, as he had the hobby of grinding lenses and combining them to produce the magnification. And he observed many natural materials under his microscope. So during his study, he has observed that raindrops contain certain tiny organism moving under the raindrop. So he called them as animalcules that laid foundation to the broad domain of microbiology, or the different organisms which are invisible to the human eye. So as I told you, it is microscopy is nothing but the study of different types of microscope, and these microscope have different types of techniques that enables us to study the organism in living condition or in a dead condition. So we can classify the microscopes into two major categories. One is a light microscope, the other one is the electron microscope. So the classification of microscope is mostly done based upon the source of light as well as the lenses that are used in the microscopy. So in case of light microscopy, the source of light is the sunlight and the lenses that are used are the glass lenses. Whereas in case of electron microscopy, the source of light is an electron beam that is coming from a cathode ray, and the lenses that are used are mostly the magnetic lenses. So this makes a difference between the light microscope and the electron microscope. Electron microscope is more sophisticated when compared to that of the light microscope. Now this light microscope is also called as optical or light microscope, and the magnification here is obtained by combining different types of glass lenses. So before proceeding with the different types of light microscope, let me explain you about the different parts of microscope that helps us to magnify the object and see it with the naked eye. So the microscope can be broadly, the parts of microscope can be broadly divided into two types. One is the mechanical portion, the other one is the optical portion. So in mechanical portion, we have all the mechanical parts such as base, we have arm, we have coarse adjustment, we have fine adjustment, we have body tube, etc. Whereas the optical portion consists of the most important part, that are the lenses that are used. The lenses that are used in light microscopy are two types, the objective lens and the other one is the eyepiece. The other parts being the condenser, etc. So the objective lens is the one that forms the primary image of the object, which is further magnified by the eyepiece. So the total magnification that we get in a light microscope is the product of magnification obtained from the objective lens and the eyepiece. So the objective lens forms the primary image, the final image is formed by the eyepiece, which can be viewed with the help of our eye. So the pathway of light that transmits itself in the microscope is from the source of light to the condenser to the object, that is, it strikes the specimen that is present on the stage, and from the stage, from the specimen to the objective lens to the eyepiece. So the final magnification that we see is a combination or the product of magnification obtained from the objective lens and the eyepiece. So these are the different parts of microscopes that helps us to magnify a particular object. Now coming to the different types of light microscope, they are classified into four types, that is bright field microscope, dark field microscope, we have phase contrast microscope, and we have fluorescent microscope. So these are different microscopes that are used to study different type of organism, like having different types of techniques. So coming to the bright field microscopy, the name itself indicates the field is bright and the organism appears dark in nature. So here the organism can be viewed either in a living form or a dead form. That is, you can fix the organism, stain the organism, and observe it under the slide. So living condition, you can observe the organism by two techniques. One is called as a wet mount technique, the other one is called as the hanging drop technique. Whereas in case of the other procedure, we have to fix the organism, we have to stain the organism, and then we have to view it under the microscope. So the specimen is prepared. Specimen is nothing but a part of the object that is usually viewed under the microscope. So the specimen is prepared and then observed under the microscope in a dead condition to study the morphology and other important components that are present in the organism. So in a bright field microscopy, the principle behind is the transmission of light. So when the organism is prepared and kept under the stage on a slide, the cone of light is passing onto the slide, and then they are taken up or they are transmitted to the objective lens, from which they are usually seen with the naked eye from the eyepiece. So here the organism have to be stained in order to bring a contrast between the organism and the surrounding. As the organisms are transparent in nature, you cannot view them directly. So you have to stain the organism with different types of biological stains. Now these stains usually brings about a contrast between the organism and the surrounding, thereby helps us to or enables us to see the microorganism which is transparent, as well as the surrounding which we are using is also transparent in nature. So by using different types of stains, we can see the different types of microorganisms under the slide. And the magnification that is obtained for this bright field microscopy is from 1000 to 2000 times. The X that represents written over there is nothing but the times. That means it increasing the object to about 1000 to 2000 times the initial object. So the magnification is obtained from the bright field microscopy is 1000 to 2000 times. Beyond this, the image becomes fuzzy. That means there is a distortion of the image. That means you will be able to see the image, but that is not very clear. And the reason behind that is because of the resolving power. A microscope should not only have a magnification but also give us a resolving power that enables us to see the clear details of the object. Now the bright field microscopy that we are using in the lab is a compound microscopy, the normal microscope that we use to study the basic characters of a particular organism. So the resolving power of a microscope depends upon two important factors. One is the wavelength of light that is used, and the other is the numerical aperture. Wavelength of the light that is used in the light microscope should be of a visible wavelength, that is the shortest wavelength of light, the greater the magnification. So the wavelength of light that is used is mostly around 300 to 400 nanometers. And next is the numerical aperture, that is nothing but the light gathering ability of the lens. That is, object should have a size of half the wavelength of light that is used to produce a good resolving power. So this is about the bright field microscopy, where you can see the organism against an illuminated background, and the organisms are usually colored to bring out a contrast between the surrounding and the organism. The next important type of microscopy is a dark field microscopy. The name itself indicates that the field is dark and the organisms are self-luminous or brightly illuminated against a dark background. So here the principle involved is the reflection of light. In the dark field microscopy, the microscope is fixed with a circular dark field condenser that usually projects the cone of light directly onto the specimens, and no other rays to fall onto the other field, that is the surrounding field. So whatever the light that is coming from the specimen gets reflected and is taken up by the objective lens. So the principle in dark field microscopy is the reflection of light. So when there is a reflection of light from the organism, it is taken up and forms the image as a bright and dark. Hence the organism usually appears as bright against a dark background. So mostly used to study organisms which are very sensitive in nature, like spirochetes. So it brings about a good contrast between the organism and the surrounding without any staining technique. So when you want to see the organism in a living unstained conditions, you can use this dark field microscopy. And coming to the next type of microscopy, which is called as a phase contrast microscopy. The principle of this phase contrast microscopy is nothing but it is based upon the refractive index differences. Every component has a different refractive index. So this is taken as a principle to produce the final image in this particular microscopy. As we all know, light travels in a particular phase, and when this phase of light strikes a particular object, there is a slight retardation in the wavelength of the light. Now this retardation in the wavelength of light brings about a phase difference between the light that has struck the organism and the light that has struck the surrounding medium. So this phase difference usually is taken as the difference and finally to produce the image in the phase contrast microscopy. So here again we have a different phase contrast condenser, and then we have a phase contrast objective that determines or that usually catches the phase difference and produces the final image. So here also you can study the organism in living and unstained condition. And next important type of microscopy is a fluorescent microscopy. And this fluorescent microscopy is based on a phenomena which is called as fluorescence. Now the fluorescence phenomena is there are certain dyes that has the ability to take up one particular wavelength of light and emit a wavelength of light which is having a longer wavelength. So this dyes takes up one wavelength of light and produces another wavelength of light, and hence they can be seen as brightly illuminated with different types of colors, beautiful colors that you can see in this fluorescence microscopy. And mostly this fluorescent microscopy is used to study immunofluorescence or immunological studies. Now next, coming to the another important type of microscope, which is an electron microscope. So the microscope, as I previously told you, can be divided into light microscope and a electron microscope. Light microscope, the differences between electron and light microscope is the source of light that we are using and the lenses that we are using. And the source of light in light microscope is a sunlight, or it can be any visible light, whereas the source of light in electron microscopy is an electron beam. And the lenses that are used in light microscopy are the glass lenses, whereas the lenses that are used in electron microscopy are the magnetic lenses. So here in electron microscopy, we have high speed electrons that are used to produce the final image, and this electron beam usually produces the image while passing through the magnetic field or the magnetic lenses. And the wavelength of light that is used in this electron microscopy is 0.05 nanometer when compared to that of the light microscope, where micrometer is or nanometer is used. Here we have 0.05 nanometer wavelength that produces more resolved image when compared to that of the light microscope. So here the resolved object is as small as 10 angstrom, and the magnification that we get from the electron microscopy is around 400,000 times that of the light microscopy. So electron microscopy is again divided into two important types. One is the transmission electron microscopy, and the other one is called as the scanning electron microscopy. And the pathway of light in electron microscopy is also similar to that of the light microscopy, where we have the high voltage gun that usually produces the electron, and then this electron passes through the first condenser lens and then passes through variety of magnetic lenses, strikes the specimen, and this differences in the intensity of the electron produced by the image that is striking the organism is finally forms a final image that can be viewed with the help of a fluorescent screen and the camera. We cannot see the final image directly with our naked eye because it can destroy the retina of our eye. So we have to first project the image onto the screen and then observe the organism. So the techniques that are involved in electron microscopy are of different types. That is, one is called as a shadow casting, the other one is a negative staining, and the final one is a free etching techniques. So these techniques help us to bring about a contrast in the organism and study not only the organism but also the internal parts of the organism. So the pathway of the light in transmission electron microscopy is similar to that of the light microscopy, where we have generation of electron from the electronic gun that passes through variety of condensers, strike the specimen, and there is a difference in the differential scattering of the electron that brings about the final image, which can be seen with the help of a fluorescent screen and the camera. We cannot view the object directly with our eye because they might damage the retina of our eye. So the image has to be first projected onto a screen and then they can be viewed. Now, there are different techniques that are used in this electron microscopy to study the organism. One of the most important technique that is used is a shadow casting technique, the other one being negative staining and the freeze etching technique. Shadow casting technique is mostly used to deposit the organism with heavy metals, evaporation of the organism with heavy metals, so that at an angle so that it forms a shadow on the other side. So the shadow is taken as an image of the object. To study the topographical structure of the organism, shadow casting technique is very important. Next, coming to the negative staining technique. Negative staining is nothing but the organisms are negative in nature, or they have a negative charge, and the dyes that are used to stain the organism, if they are negative, the negative repels, and this dye, instead of staining the organism, they will stain the background. So this concept is mostly used to stain the background to bring about a contrast between the organism and the background. So here we have the heavy metal acids that are used, which is called as a phosphotungstic acid, that usually deposits on the on the background of the organism to bring about a contrast and to study the organism and the electron microscopy. The next part is a freeze etching technique. So in case of electron microscopy, we have to prepare ultra thin section of the organism. In that case, we are completely killing the organism. So we cannot study the organism in living condition, neither we can study the internal structures of organism. So the best method that is used to study the organism or the internal parts of the organism is a freeze etching technique, where the technique that is involved is using the organism which have been defreezed in a gas, liquid gas, and then cutting the organism or etching the organism with the help of a diamond knife that has been already freeze. So this opens up the molecule, and carbon replicas are made which can use to form the final image of the internal components that are present in the organism. That is the logic behind the freeze etching technique, that helps to study the organism by creating a etch with the help of a diamond knife to open up the organism and then see the internal parts of the organism without distorting any particular object inside the organism. Another important type of electron microscopy to study the three-dimensional structure of the organism is a scanning electron microscopy. So the scanning electron microscopy depends upon two importance of electrons. One is a primary electron that showers on the organism, which produces differences in the differential scattering of the electrons that are taken up by the detector and finally forms the image on the fluorescent screen. So scanning electron microscopy helps us to give the three-dimensional structure of the organism and the compounds that are usually studied under that particular microscope. Now, this is about the microscopy that we have studied. That is, without this microscope, we will not be able to know the invisible domain of microorganism. There are different techniques that are usually seen in combination with the microscopy. One is the important is the staining technique. Staining is nothing but to improve the contrast between the organism and the surrounding. So different stains that are used to study the different types of organism. There is a gram staining or differential staining that helps us to differentiate bacteria into gram positive and gram negative. There are certain components of the organisms that cannot be stained directly with the normal dyes, so we have to go about the specific type of staining techniques that includes the acid-fast staining or Zeil-Nielsen staining. They include endospore staining, etc. So there are different stains that are used in microbiology to fix the organism, stain the organism, and study the organism under the microscope. Coming to the shape of the bacteria, the most important part of the morphology of bacteria, these bacteria can either be in the form of spherical or round, which is represented as cocci. They can be in the form of a rod, that is represented as bacilli. They can be in the form of a curve, slightly curved, which is nothing but the vibrio. They can be rigid spiral, which is nothing but spirilla. They can be flexuous spiral, which are nothing but the spirochete. And the other structure or the shape of the bacteria can be star or square. So apart from that, we also have other type of bacteria which are called as actinomycetes. They are resembling the fungus in nature. That's why they are called as actinomycetes. And the next shape is of mycoplasma. These are the organism without the cell wall. So these organisms are bacteria with different types of shapes. Coming to the arrangement of the bacteria, these bacteria arranges themselves after the plane of division. The cocci, if they are arranged in twos, they are called as diplococci. If they are arranged in a chain, they are called as streptococci. If they are arranged in bunches, they are called as staphylococci. If they are arranged in tetrad, they are called as sarcina, etc. Similarly, we have bacilli that arranges themselves. If they are a chain of bacilli, then it's called as a streptobacilli. If they are arranging them in a plane, this form is called as a cuneiform or a Chinese letter form, mostly seen in Corynebacteria. So this is about the shape of the bacteria that helps us to identify and characterize the bacteria. Now coming to the bacterial morphology, the most important components which are present within and outside the bacteria. So I will classify this bacterial component mostly into three. That is cell wall and components outer to the cell wall and components inner to the cell wall. Coming to the components that are outer to the cell wall are flagella. We have fimbriae or pili, we have glycocalyx that includes the capsule or the slime layer. And components inner to the or internal to the cell wall are the cytoplasmic membrane, cytoplasm, and the components which are present in the cytoplasm such as ribosomes, we have nucleus, we have intracytoplasmic granules, we have the other components such as spores, etc. So first important components that is external to the cell wall is the flagella. Flagella is a locomotory organelle that brings about a motility in the organism. So they are present in most of the organism but not in all organism. The organism which have this particular flagella is called as a motile organism, and this organism moves based upon the chemotaxis. So whenever they find a chemical, they move towards it with the help of the flagella, and this procedure is called as chemotaxis. And these flagella are fine thread-like appendages that are seen external to the cell wall, mostly measuring about 20 nanometer in diameter and 15 to 20 micrometer in length. And the parts of the flagella is usually consist of three important parts. One is the flagella, which is external, and then it is connected to the hook, and which in turn is connected to the basal body. So the flagella or the filament and the hook are usually present on the cell surface, whereas the basal body is usually embedded in the cell envelope. So this flagella or the filament ends in a square shaped tip, and in order to see the flagella under the microscope, it is very difficult. So you have to increase the diameter of the flagella and then study it under the microscope. So that particular staining method is called as using a mordant. Mordant helps to fix the flagella and increase the diameter so that we can stain with the different stains and then view it under the microscope. So the bacteria usually are motile in nature, and they have different types of flagella. You can see the different types of flagella over there. So the flagella can be present throughout the whole organism, which is called as a peritrichous flagella. And if there is a single flagellum present at one end or both the end, it is called as a polar flagellum. And if there are tough flagellum present at one end, it is called as locotrichous. And if they are present at two ends, that is a single flagellum, they are called as bipolar flagellum. So there are different arrangement of flagella on a microorganism. So they can be present throughout the whole microorganism, which is called as the peritrichous flagella, usually seen in the typhoid bacilli, and then you have a single flagellum present at one end or both the ends. So they are called as polar flagellum. Is a tuft of flagellum present at one end, it is called as lophotrichous. The single flagellum present at one end, it is usually seen in cholera, and then if the seen tuft of flagellum or lophotrichous is mostly seen in the spirilla bacterium. And coming to the determination of the motility of the organism, they are difficult to demonstrate under the microscope. So we use a media to demonstrate this motility of the organism. So they can be seen on a solid media, either in the form of a stately, usually seen in bacilli, or in a darting movement that is seen in a Vibrio organism. So this is the most important locomotory organ, and the protein with which the flagella is made up of is called as flagellin. Now coming to the next important appendage of the microorganism is a fimbriae or the pili. Fimbriae or the pili are the adherence appendages. These are usually seen in some of the organism, again not in all organism, mostly seen in gram-negative organism, and they are non-locomotory. So they can be present in both motile as well as non-motile organism, and they are more fine and hair-like or thin when compared to that of the flagella. And the presence of this flagella usually forms pellicle or comes in a liquid medium. So the flagella are mostly the attachment appendages. They attach to different components in the surrounding media. So these flagella also have the ability to attach to the RBCs. So they are called, this procedure is called as hemagglutination reaction. So helps us to identify the organism when they are mixed with the RBCs of certain types of organism. So they go and bind to them, they form the hemagglutination. Depending upon the hemagglutination, you can identify or characterize the bacteria. And the other important type of the flagella, the fimbriae is the pili. Pili is made up of a protein which is called as pylin, and this usually helps in the conjugation. That is, the organism, the gram-negative organism having pili is said to be a male organism which have the ability to donate or transfer its genetic material to the female organism, which is represented as F minus. So when the F plus mates with the F minus, there is a conjugation tube that is formed, helps in transfer of the genetic material from donor cell to the recipient cell, and which helps to convert the female cell also to the male cell, which again can further transfer the genetic material to the other cell. So they helps in conjugation, that is transfer of genetic material. Next important appendages seen in most of the organism again is a glycocalyx. So glycocalyx is a viscous material that is surrounded surrounded on the organisms and secreted by the organisms, that helps us to protect itself from the external environment. So glycocalyx can be in the form of organized structure which is called as a capsule, or it can be in a disorganized loose glycocalyx which is called as a slime layer, and mostly made up of glycoprotein or the polysaccharides. And glycocalyx or the capsular material that is present in the organism makes the organism more virulent when compared to the other organism. Take for example pneumococci. So these organisms have a capsule around itself. So they usually escape the phagocytic cells that usually engulfs the organism and eliminates the organism in the immune system. So these organisms with the capsulated material escape phagocytosis, thereby increasing their pathogenic character. So the capsules are the most important part. They cannot be easily stained with the normal stain. You need to have a capsular staining technique where copper is mostly used that acts as a mordant. And encapsulated organisms are more pathogenic and virulent when compared to that of non-capsulated organism. Next, coming to the important component which is nothing but the cell wall. Now the bacteria has a cell wall that helps us to give its rigidity, a ductility and a shape. The cell wall of the bacteria protects the organism from the external environment and protects the internal component of the organism. Now, when there is a rupture in the cell wall, there is a death of the organism either due to the leakage of the content or due to the osmolysis. Now, the cell wall of the bacteria is more complex when compared to the other organism. So, based upon the chemical composition, you can classify the bacteria into two types, gram-positive bacteria and gram-negative bacteria. Basically, both the bacteria are made up of a similar component which is called as peptidoglycan layer or a murine layer, which is nothing but a mucopeptides. And the peptidoglycan layer is made up of two important sugars. One is called as the N-acetyl glucosamine, the other one is called as N-acetyl muramic acid, which are linked together by a bridge which is called as beta-1,4-glycosidic linkage. And one sheet of peptidoglycan layer binds with another sheet of peptidoglycan layer with a tetrapeptide linkage. So the chemical composition again varies from one organism to the other organism. So cell wall is the most important part of the bacteria, and bacteria can be broadly classified into gram-positive or gram-negative. This cell wall is mostly made up of the carbohydrates, the lipids in some cases of bacteria such as mycobacteria. They also have an external lipid component in the cell wall. So these mycobacteria have to be stained with different types of staining technique that is called as Ziehl-Neelsen method, whereas the normal bacteria can be stained with the help of Gram staining. It was Christian Grams in 1884 discovered this particular phenomena, staining technique which is called as Gram staining, helps to classify the bacteria into Gram positive and Gram negative based upon the chemical composition of the bacteria. Now, the gram-positive bacteria and gram-negative bacteria differences is that gram-positive bacteria is very simple when compared to that of the gram-negative bacteria, and it is more thick, having more layers of peptidoglycan than that of the gram-negative, which is thin in comparison to the gram-positive bacteria. And the important component of gram-positive bacteria is the teichoic acid, whereas in gram-negative bacteria we have a specialized layer which is called as LPS layer, that is nothing but lipopolysaccharide layer. So the lipopolysaccharide layer is very important part of the component in the gram-negative bacteria, having three important parts. One is the O antigen, you have a core antigen, and then you have a lipid A portion. The lipid A portion usually usually counts for endotoxicity of the organism that brings about the fever-inducing property in a gram-negative bacteria or tumor necrosis factor of the gram-negative bacteria. So this gram-negative bacteria are more complex when compared to that of the gram-positive bacteria, having O antigen, core antigen and lipid A portion. So gram-negative bacteria usually have an external layer which is called as lipopolysaccharide layer, having three important components: O antigen, core saccharide and lipid A antigen. So this lipid A antigen is the one that has the endotoxic activity, that is pyrogenic in nature, that is fever inducing object in an organism. So whenever this organism infects a particular organism, they usually causes fever. So there is an increase in the temperature of the body and regulating the hypothalamus region. So lipid A region is the one that usually contributes to the pyrogenicity or inducing fever. Apart from that, it also acts as a tumor necrotic factor and other endotoxic activity of this lipid A region. So this is a main difference between the gram-positive bacteria and gram-negative bacteria, where gram-positive is more simple when compared to that of the gram-negative bacteria. And apart from this lipopolysaccharide layer, certain bacteria also have an outer membrane proteins. So they are called as OMP proteins, having porins. So they help to translocate a small molecule in and outer to the external environment, and they are the region where the bacteriophages usually attacks the bacteria. Bacteriophages are nothing but the viruses which attacks the bacteria. So OMP proteins are the one that helps to bind the bacteriophages. So this is about the biochemical nature of the bacteria, which is made up of peptidoglycan layer and differences of gram-positive and gram-negative bacteria. Now coming to the structures internal to the cell wall, which is nothing but the cytoplasmic membrane, inside to which is a protoplasm or the cytoplasm having different inclusion bodies and the different components inside the protoplasm. So coming to the cytoplasmic membrane, it is also called as a unit membrane, and it has a function of semi-permeability, able to transport only few things inside and outside to the external environment. Semi-permeable membrane helps the bacteria not to take up all the material that is from the external environment, not to exit all the material that is internal to the environment. So it has a semi-permeability, allows only certain components to enter into the cell and away or outer to the cell. So this is called as a unit membrane having three layers. That's why it is called as a trilaminar unit membrane. And the model that was proposed is a fluid mosaic model, and it is made up of carbohydrate, proteins, and in some cases of bacteria such as mycobacteria, they also have sterols in them, and mostly thin in nature, having 5 to 10 nanometer thick. Now coming to internal to the cytoplasmic membrane or the semi-permeable membrane, it is a cytoplasm. It is a colloidal system having different components including organic, inorganic molecule plus water, and they do not show any cytoplasmic movement that is seen in eukaryotes. And the components that are present inside the cytoplasm are ribosomes, you have nucleus, you have plasmids, you have intracytoplasmic granules, as well as certain organisms also do have spores inside them. So coming to the ribosomes in bacteria are of 70S type in comparison to the eukaryotes, which are of 80S type. S is a sedimentation unit in the centrifuge. So ribosomes have 70S type of ribosome in case of prokaryotes, that is 30S and 50S, that helps in protein synthesis. Now coming to the mesosome, it's nothing but the invagination of the cytoplasmic membrane inwards to the cytoplasm. It acts as most of the respiratory enzymes are usually present in that, helps to carry out respiration in microorganism, whether it is an oxidative phosphorylation or a substrate level phosphorylation. It is the most important site where the respiratory enzymes are active and carry out the respiration process. Now next coming to the nucleus has as such there is no nucleus in the microorganism, that is these are called as prokaryotes because they have primitive nucleus. So there is a nucleolus rather than a nucleus, and this nucleolus has a single form of double-stranded DNA molecule without any type of proteins attached to it as seen in the eukaryotes. Now the other components apart from nucleus, we also have plasmids, which are nothing but the extra chromosomal materials, and these extra chromosomal materials are the one that carries certain types of resistance factors inside them. So the organism becomes resistant to different types of antibiotics because of the R gene that is present in the plasmid. So nowadays we have got different organisms that are called as multiple drug resistant organisms because they have different resistant factor genes on the plasmid. So it is the organism that becomes resistant but not the human beings towards particular antibiotic. So plasmids are the extra chromosomal material, that is apart from the normal chromosome that is present in the bacteria, there is an extra chromosome that is present in the organism is the plasmid. Next important components seen in the cytoplasm are the intra-cytoplasmic inclusion bodies, that includes volatile granules, polysaccharide, gas vacuoles, etc. So volatile granules are also called as Babes-Ernst granules, and these granules are nothing but the storage of certain kinds of food, and they can be demonstrated with the help of different staining techniques. So present in only few species of the organism, and they help to characterize organism depending upon the volatile granules that are present. Now coming to the polysaccharide granules and lipid granules that are seen, polysaccharide can be stained with the help of starch, and the lipid granules can be stained with the help of fat soluble staining that are called as Sudan black. And finally gas vacuoles, they are nothing but the storage of certain types of phosphate energies so that the organism can carry out metabolic functions. Now the last important component seen in the organisms are the spores. Spores are produced in certain types of organisms such as Bacillus and Clostridium during unfavorable condition. So when there is a depletion of nutrition in the surrounding media, the organism produces certain types of spores that helps them to survive for a longer period of time till the return of a normal environment. So these organisms which has developed into spores can survive as long as 4,000 to 5,000 years together. So as soon as there is a return of the normal environment, these organism produces a vegetative cells and carry on its normal function. So you can say that these spores are nothing but a type of reproducing bodies in the organism. Spore from the vegetative cells are formed, and this on return of natural condition can also generate into vegetative cells. So seen only in few of the organisms have the ability to produce the particular spores. As these spores are produced inside the body, they are called as endospores, and the process is called as sporulation. And these spore cell has different layers. It has a nuclei, it has a core, it has a spore coat, okay, and this spore coat helps to protect bacteria in unfavorable condition, protects it from desiccation, protects it from high temperature, protects it from heat, etc. So the organism are more resistant forms seen in the sporulating forms, and these can be stained with a special type of staining technique which are called as endospore staining technique, where malachite green is used, and this organism are exposed to steam so that the external layer becomes permeable, so that the organism takes up the primary stain and stain the spores. So once the spore takes up the stain, it will not leave the stain, and you can see it under the microscope as green colored against a pink background. That is, the vegetative cells are counter stained with safranin and the spores are stained with malachite green. So you can see these endospores very beautifully as green in a vegetative form. So this is all about the morphology of bacteria.